All right, everyone. Well, thank you for staying here, and we are excited. We definitely don't want to say save the best for last, but we have a large interest in our region about hemlock woolly adelgid, HWA, and we are very fortunate to have Dr. Carl Fetter here. He is a plant biologist working in conservation genomics, quantitative genetics, and genomic methods development. So his research interests have coalesced into a research program that bridges the gap between conservation, breeding, and advanced genomic methods. So we're really going to get some good insights about what are some of these new tools that can help us protect our hemlocks. So can everyone, you got me, give it a round of applause. Thank you.
Okay, I am the last speaker. Glad to wrap this up with some optimism. Of course, with conservation, there's lots of challenges, but there are many of us that are in the room and are also not in the room that are working toward solutions. I want to give a big thank you to Tamara Van Ren for inviting me in April at a meeting we were both at. It was really fun to get the invitation to come here. I did my PhD at the University of Vermont, and I was there for 5 years. I left Vermont, went to Virginia, went down to Georgia. And today, when I got the blustery, cold wind, I loved it. I was like, oh, this is so good to be cold again. It's been a while since I've been cold. Forests are obviously really important. I don't have to convince people in this room that they're important. They're important for cultural reasons, economic reasons. They're just, critically important for our lives and modern society. Unfortunately, a lot of trees are facing problems that are due to invasive pests and invasive pathogens. So here are just a couple examples of trees that I think about that are facing different threats from the hemlock woolly adelgid. So this is an invasive pest from Eastern Asia, from fungi like Ophiomyia that affects butternuts and will kill the trees in just a few years. And then American chestnut is a really well-known tree that is died from chestnut plague. So globally, what this situation looks like is that about a little more than 30% of trees globally are threatened or facing some kind of a population collapse. In the U.S., this number is about 12% of species. So that's a lot of trees that are facing conservation issues. So conservation genomics, this can mean different things to different people. And when I think of conservation genomics, I have in my mind a program that involves different people, different organizations, different skill sets to perform conservation that is enabled by genomic technologies. So for me, this often starts with reference genome sequencing. So I have a couple slides that introduces that more in a moment. Going back into the landscape and doing population resequencing or population sampling to get an understanding for the genetic diversity that's present in the species. Doing some kind of deployment of those of germplasm in a common garden or in some kind of controlled environment so that you can start to do quantitative genetic modeling. Quantitative genetics, by the way, it's a wonderful technique. Really, it doesn't require any sequencing whatsoever. So it's really accessible if you have trees in the ground or organisms that are reared in a common environment. It's just ANOVA. ANOVA was actually invented for these methods. So really kind of first way for a lot of people to get into breeding. And then another part, a critical component of conservation genomics is community outreach and infrastructure building. So these, where are my arrows? These different spheres of working interact with one another and create this feedback loop that, in my mind, is what the conservation genomics is all about. So I'm in the plant computational genomics lab at the University of Connecticut. I'm a postdoc there. I'm funded through a USDA postdoc fellowship there. And here are just some of the species that we work on. So we're working on Eastern hemlock, and we have sequenced the genome of the tree and its pest. We're working on butternut, which is Juglans cinerea. So butternut is native to the East Coast and is an endangered species. Japanese walnut is an introduced species of walnut that are hybridized with Dakota nuts, and it's largely resistant or tolerant to the fungus, so we're working on that one. We're also working on other cruel trees of conservation concern. The Florida nutmeg tree, Torreya taxifolia. It exists in 2 counties globally, one in Georgia and one in Florida. And then we're working on other variety of cool species there. We're doing population resequencing and population work in eastern hemlock, butternut. We're starting work in the Carolina hemlock and also red bays, going to be a new focus for us. For quantitative genetic modeling, this is just a little bit of what I'm actually working on. There are other traits that we're working on in the lab, but it's quantitative genetics is all about trait modeling. And then for community outreach and infrastructure building, this is something that is driving some of our projects and is a result of what we're trying to do. So I've been working on the hemlock for about a year and a half or so, and it's been one of the most fun species I've ever worked on because everybody loves hemlocks. If you call somebody up and say, hey, you take Tuesday morning, go in the field, collect some leaf tissue from me, it's always a yes, because people just love the trees so much. It's been fun to build relationships with land managers and to think about ways to deploy what we're doing to get seed collections and seed banking. Another important component of what we're doing is a training program for students through the Biodiversity and Conservation Genomics Center. And another important partnership that we developed in the lab is with Org1. So Org1 is this nonprofit that's funded by Oxford Name for Technologies, which makes sequencers. And Org1's goal is to sequence the genome of every critically endangered or endangered organism on Earth. In the United States, IPI's lab is the U.S. Sequencing Center for Org1. So we work with people from all over the country, different museums, different laboratories to extract their tissues and do protein-based sequencing. So that's been a really fun program. But this for me is the kind of conservation genomics program that works for trees. So a genome, what is a genome? I have a couple slides to just kind of introduce a little bit of concepts in genomics. So a genome is the aggregation of all of the sequence coding information in an organism. Here, this is a karyotype of the chromosomes from Juglans cinerea. So we have 16 chromosomes here, 16 pairs of chromosomes here, one for mom, one for dad. What's not represented here are the plastids, so the chromosomes not represented here. But all of this information here is kind of what we're trying to capture with the genome sequencing project. There's lots of other information that is epigenetic, like patterns of methylation, that are also, I think, increasingly considered part of the genome. And depending on your technology, you can sequence information about what's methylated, what's unmethylated, different types of methylation that are present. So to get a genome sequence, we're going to start with some kind of a tissue. Depending on your organism, you're going to take different approaches for doing an extraction or cleaning up the DNA, getting rid of stuff you don't want. This is a little cartoon of a Promethean sequencer, and then you get raw reads. So you're going to get millions and millions and millions of reads, and you have to do something with those reads. That they're going to be, you're going to have adapters in them, you're going to have parts of what you want, things that you don't want. You're going to have overrepresentation of some regions, underrepresentation of other regions. So you're going to have to kind of figure out ways to assemble this to get it to work. So here's kind of just a basic cartoon of genome assembly. So you get your raw reads up top. Like I said, you have millions of them. They're going to be unequally distributed. Those are assembled into contigs, which can be hundreds of thousands of base pairs long. The sequence reads can be 150 base pairs, or 50 base pairs, or 300 or 550 if you're doing Illumina technologies. With Nanopore, they could be tens of thousands of bases long. But you assemble them into contigs. Those contigs are then assembled into scaffolds, which may have some number of missing sites indicated by these ends that are present there. Your hope is that you have at least as many scaffolds as you have chromosomes. It doesn't always happen, but sometimes you get lucky and it does. And then you'll have a number of small scaffolds that you just kind of throw out. So once you get a genome sequence, hopefully you have it at least at some chromosome level. You need to, it's just a bunch of A's, T's, and G's, and C's, and N's. So in order to get some useful information out of that, you have to annotate the genome with structural gene information about where genes start and stop, and then functional information about what those genes are. So here I've just kind of represented a cartoon of what a gene might look like with a UTR, exons, introns, 3' UTR. And then after it's annotated, you're going to get a file format like this that tells you there's a gene that's here, there's a start codon that's here, positional information of it. If anybody is interested in this type of work, this genome annotation, this is really a whole field into and on itself. And my advisor, Dr. Jill Wegerson, and labmate Cynthia Webster developed this tool called Easel, which is a genome annotation pipeline that is working really well for organisms. So after you have your structural annotation, you want to functionally annotate it with different tools. So basically, you're just comparing what the gene levels you found to databases of other known genes, and then you functionally annotate them. So reference genome projects, the goal is to have the assembly and the annotation, right? But along the way of doing that, you kind of create all these other amazing relationships and opportunities for collaboration. So if you're ever thinking to yourself, what's the use of one more reference genome? Well, really, it's one more project that allows you to create a system for doing conservation. So if you want to get tissue, you need to work with arboretum, botanic gardens, breeders. There are training opportunities in wet labs for undergraduates, for rotation students who work on these projects. The sequencing facility, there's a lot of people that are working in there that are your partners. So genome assembly and annotation is its own area. And then all of this information is useful for geneticists, breeders, evolutionary biologists. So these projects are really fantastic ways to bring together communities of scientists and non-scientists and land managers. And that's one reason why I'm so excited to work in this area. And community partners. So just a little bit about one program that we have going on at the University of Connecticut. It's the Biodiversity and Conservation Genomics Center. It's in the Institute for Systems Genomics. And this is a program that a couple of PIs have come together to create. And basically, we take smart, motivated undergraduates that have never done anything with genomics, and we train them through this kind of paradigm of conservation genomics. So we always have a joint project that everyone works on. This was the butternut genome project. So we did a genome sequence of this one, of a butternut session in Canada. Everybody has one joint project, and then there are various parts of the projects that people specialize in. So the students get a lot of exposure for different skill sets. And then they're paired with graduate students. So Michelle and Cynthia are graduate students in the program. So is Ame. And then I was a postdoc with this cohort. And then Jill and Rachel are the other two PIs. So really, again, just amazing to see all this come together. And this paper, I'd say, was 90% driven by undergraduate thinkers doing the work. It was just us kind of directing them in this direction. So really fun to do that. So in my lab, the lab I'm working in, working on the Eastern Hemlock and butternut primarily. And so today I'll tell you a little bit more about Eastern Hemlock. It's listed as near threatened by the IUCN. So here's the team. Again, this is my PI, Jill Wegerson. This is Cynthia. This is Vidya, who's done a ton of work on this. This is me with the Hemlock Restoration Initiative in Asheville, North Carolina last year. So just again, a great group of people to work with. This is Chris Gruzman, who's a first-generation college student who's from a family from the Dominican Republic. So he's the first person to go to college and to work with him and other students who this is something new for them. It was an important part of conservation. Okay, so this is my introduction slide for the Eastern Hemlock. We have a wonderful stand of hemlocks here along a creek. I was at a hemlock art exhibit in Huntsville, Alabama last year, and I saw this poem. And after I read this poem, I thought, oh, this is great. This kind of covers all the points of hemlock, of an introduction to hemlock biology. So it's a threatened tree. The tree itself is important for conserving and preserving diversity. There are lots of other species as warblers and wrens and grouse. It has important ecological roles for preserving the abiotic environment. It's important. It's culturally important for Native American communities and anybody that loves to go into an environment like this. So yeah, that's my hemlock introduction. Here's the range of it: classic Canadian funnel distribution, and I think probably everyone knows about this. So hemlock woolly adelgid. It is an invasive pest from Eastern Asia. It occurs in Japan and kind of Japan, China, those areas. It was imported into Richmond, Virginia in the 1920s, at least in the 1920s, a couple times. In the 20s, it was fashionable to have Asian gardens, and there was a prominent family in Virginia that brought over Subus eboldiae, the southern Japanese hemlock, and planted it in their Asian garden. This is their, I guess, homestead is now a city park in Richmond, Virginia. It's a Maymont Park. I used to live in Richmond and would walk around there. And so it's kind of been on my to-do list to talk to them and see if they have records about what was planted where, and to see if we can find the actual bed where it was planted, and maybe put some kind of informational sign there. But started in Richmond, it spread out. The adelgids are hemipterans, so they're true bugs. They are sap-feeding, and they insert a stylet at the base of the petiole. And it's thought that they kill the hemlock tree by initiating a hypersensitive response. So this is kind of like the tree gets a fever, it gives itself a fever. And hypersensitive responses work pretty well to eliminate lots of pests and pathogens. But it's if you have imagine having a fever for 4 to 8 years or so, eventually you're going to succumb. So that's what ends up killing you. So the program, the conservation genomics program that we envision for this species has a couple different names to it. It starts again with reference genome sequencing of the host and the pathogen. We then move on to comparative transcriptomics and metabolomics of different species in the hemlock genus. There are about 10 different species, and they have differential response or differential tolerance to HCBA. We then rely on some pre-existing experiments to do some within-species comparisons of HCBA tolerance and terpene, and try to see if there's some kind of a ecological role for terpenes. And finally, the fourth aim of this is climate adaptation. So doing landscape genomics to identify potential regions of the genome that are adapted to climate, and then use that information to support seeding. So the Nature Conservancy and the Tree Species in Peril program was really important for providing funding and innovation and communication for several of these aims. And then my USDA postdoc provided funding for the first and fourth. Okay, so our reference genome. This is the genome tree at the order of Arboretum. It's a huge prominent hemlock that's right along one of their kind of main paths into the Arboretum. So if you go into there and just follow the main path, you'll see a giant hemlock tree going over the path. That's the genome tree. It is a local Bostonian tree that was grafted from a local plant and then planted there. So in order to get DNA sequence, we use a technique to obtain nuclei and then release those nuclei. And the nuclei are the little blue dots that are there. Hemlocks do this fun thing where they put condensed tannins into the chromatin, which is really annoying if you're trying to get clean DNA. So we had to do quite a bit of troubleshooting to get reads that were clean enough to see, or DNA that was clean enough to sequence. The estimated genome size for the species was anywhere from 18 to 21 gigabases. The human genome is 3 gigabases. We're doing about 18 gigabases. So quite a bit of sequencing. We had to have 10 high molecular weight libraries constructed for 10 Prometheon nodes. And so Prometheon is similar to this Minion sequencer, except it's much higher throughput, and it will generate about 100 times more data than Minion will. And so we had to have 10 of them go in. And we ended up generating about 1.6 terabytes of sequence data. So it was quite a bit. We also did Illumina short read sequencing, which we were potentially going to use for error correction. So we assembled it, we removed small fragments, A lot of times with ONT assembly, there's higher error rate, so we would bring in the short reads, which have a lower error rate, or correct them. It turns out actually that right now the ONT reads are good enough that our assembly was better quality without them than with them. So the hybrid assembly was not favored, and we just use an ONT only assembly. This is our Busco score. So Busco is based off of the Spanish word buscar, to search. So when you do if you want to know how complete your genome assembly is, you do a Busco search. And what you're doing is there are about 1,600 proteins that should be present in all organisms of a certain clade. And if you have a good assembly, when you go in to search for those 1,600 proteins, you should find them. So if you have a 100% score, at least all of your Busco genes are there, and that gives you confidence in the assembly. We have a Busco score of 81, which for a conifer is very good. So we were quite happy with this. We sent off some sample to Contacta Bio, which is a biotech firm in San Diego, to do high-C sequencing. So I will show you what this looks like. Okay, so here are high-C sequencing results. Basically what this is a map of cross-linking between regions of single chromosomes. Well, you've got this kind of diagonal structure here with these 12 boxes that are there. That what you do is you get your DNA, and then it's cross-linked with a molecule, and then it's broken up and sequenced. And so when you have regions of chromosomes that are cross-linked and touching one another, that's because they're homologous regions of chromosomes. So you could use that positional information to reconstruct a chromosome map, and that's what we've done here. So after we did the high-C sequencing, our Busco score went up about 10 points to 91, which is really fantastic. We're really pretty happy with that. So we have a chromosome scale assembly for this retinol. This is fantastic. Next steps are genome annotation. We have RNA libraries available to do genome annotation. The RNA libraries tell you what your transcripts look like, and that helps EASL and other tools annotate the genome. So that's the next thing that's coming. In the meantime, we wanted to sequence the genome of a pest. So I went to the Mountain Research Station, which is in Waynesville, North Carolina, and collected some clonal individuals for sequencing. We used one ONT library to sequence them. The genome size is about 230 megabases, and so 99.9% of the sequences were contained in 9 scaffolds. So we got the chromosome scale assembly without having to do high-C or any additional sequencing, which is really quite nice. Good Busco score of 98%. So this work was largely conducted by Adam Glendenning, who is a RAMP student. So RAMP students are kind of like postbac students that come back to a particular lab to do some research. So Adam did a lot of this work. And along with Tim Cernak at the University of Michigan, they're interested in studying nicotine receptor proteins to try to see if they can develop other methods for pesticides and different natural pesticides. So this genome is going to be published soon. We've already sent it out to some collaborators who are interested in RNAi interference work. So Andrew Groover, who's at the Northern Research Station, U.S. Forest Service Northern Research Station, located in Burlington, Vermont. He has started a new project to do RNAi on 5 or 6 different pests. So ashes is one of the species, Adelgid, and a few other that I can't quite remember. Dana Nelson at the University of Kentucky, also Forest Service, is going to be leading the Adelgid work there. So it's going to be really, really amazing to see this come together and some potential biocontrol, or not biocontrol, potential RNAi control. Okay, so our next aim is to look at different patterns of metabolomics across species in Sudan. So just a little review about terpenes. So plants, obviously, they can't move. They don't have to be able to avoid problems, but they do have this immense toolkit of metabolites and things that they sequence. Terpenes are a highly diverse set of chemical compounds that are synthesized by plants. There's about There's more than 30,000 of them. The basic building unit of a terpene is an isoprene, which is a 5-carbon ring that's represented here. These are terpenes are ways, in other metabolites, are ways that plants deal with environmental stress. It's pretty well known that terpenes are signaling molecules between plants and insects. So, for example, in cycads, there we go. In cycads, I was just at a talk about 2 weeks ago. It was really cool. Cycads, they're pollinators of beetles, and they synthesize 2 different terpenes in order to communicate to the beetles that the male organ is ready to be for them to come in, get pollen, and then go to the female cone. So these chemicals are signaling chemicals, and they're doing a lot of interesting ecological work. So knowing that, thinking about this, we wanted to try to investigate an ecological role for them. So let me get your attention over here first. So this is an experiment that compares the amount of hemlock woolly adelgid on 4 different species of hemlock. So we have our eastern hemlock here, which is largely susceptible, our Chinese hemlock, which is largely tolerant or resistant to the adelgid, and then 2 other Asian species. So in these laboratory inoculations, you can see that there's a lot more adelgid on the canadensis than the chinesis. So if you just compare total terpenes, this is from a different experiment. This is something where we went back to the Arnold Arboretum and we collected tissue. But just right off the bat, if you just compare this, you see high terpenes, high adelgids. When you compare it with chinensis, we have lower terpenes. This is from 6 different terpenes that were all aggregated together. Fewer adelgids that's there. So it kind of gives us a very rough thought. Perhaps this is an avenue that we should investigate more. Another interesting thing, a pattern that I look at, so I did most of my training as an evolutionary biologist, so when I look at these different species, I ask myself, why is there such a huge discrepancy in the amount of HWA that's present on these different things? And obviously, canadensis didn't evolve in the presence of these adelgids. So that's kind of the ultimate reason, but mechanistically, what's going on? To try to investigate this, we wanted to look at the evolution of terpene synthesis. This work was started before we had a genome, so we would not necessarily do it this way today. But we gathered a bunch of transcriptomes from things that we had sequenced and also from CBI. We assembled these transcriptomes across many different conifers, and then we started counting the number of terpene synthases that were present. So if you just look at the hemlock group, Suga is up here from Mertensiana up. There's no real great signal that says, there are more terpene synthases, and that's why there are fewer terpene synthases, whatever. No great signal that's there, unfortunately, in terpene synthase count. When we build a tree from these terpene synthases, it's just kind of a sanity check to be sure what we have. We are recovering the correct topology that should be there. A lot of the conifer terpene synthases are in this terpene synthase D clade that's over here. And so this study is kind of where it's at right now. We want to go back into this and look at the sequence evolution of binding sites to see if there's somehow different regulation of these terpene pathways. But this is something we're going to come back to. So the next aim is to look at within species patterns of resistance and terpenes. So I think many people have probably heard about the bulletproof stand, so the bulletproof trees that come from New Jersey. Evan Pricer and a graduate student planted, so they got cuttings and open pollinated seed from the bulletproof stand and planted them in 8 different common gardens along the East Coast. And they planted them in, I think about 2010 was when they were planted, and came back, counted adelgid counts, and said, okay, there's a difference between how many delicate are settling on these bulletproof trees and how many are settling on controls. So we wanted to go back into these stands and measure terpenes to see if there was a role there. In the meantime, we have a little bit of kind of interesting data here. So you can see that there's a site effect here. Here's our Pennsylvania common garden, our North Carolina common garden. You can see overall there's a lot more adelgid that is in the Pennsylvania garden than the North Carolina garden. But if you compare what your expectations are for bulletproof versus non-bulletproof, in Pennsylvania, there's actually more adelgid that's settling on the bulletproof trees than in the non-bulletproof trees. And, yeah.
I just want to mention, I saw some people not nodding their heads about the bulletproof trees. So just a quick mention that New Jersey did about a 20-year study of a lot of different hemlock stands and how many trees were surviving. And they kind of stumbled upon literally this one area of hemlock trees that appears to be resistant to hemlock bulletproof. So where all the other trees were dying, they were still able to have 80 or 90% of the trees of this particular tree live, even though there's hemlock bulletproof all over. And so they have become the bulletproof stand, and they're like the holy grail.
Yeah, it's very much a holy grail type situation where it's hard to get in there to study them because they think they have something that's so special that no one can touch it, which is kind of funny. The bulletproof is it's definitely not bulletproof, right? That's what these data are showing. In North Carolina, you kind of you do recover a signal where the non-bulletproofs have more delta than the bulletproofs. But I'm going to show some data in a moment that shows you what the real effect is going on here. So sample size is this differential number of trees. Okay, so terpenes across our site. So these are total terpenes. We measured 10 different terpenes, some monoterpenes, some sesame terpenes. We aggregate their numbers together, and these are some interesting signals that we get. First, look at the Arnold arboretum. You can see, and our two different colors here are bulletproof versus non-bulletproof ones. First thing that pops out to me is this seasonal pattern of increase in terpene expression that we see there. So there's a peak around April, and there's another peak around July, and then there's this fall peak. It was suggested in a 2013 paper that there was a difference in the timing of, a difference in the amount of terpenes that were produced in the bulletproof and non-bulletproof lines, and that may have something to do with resistance patterns. Remember I told you that more terpenes were correlated with more adelgid in our cross-species study. But we do recover this difference between bulletproof and non-bulletproof. Same thing here. Here we've got the non-bulletproofs that are making more, but not in non-bulletproof. So one interesting just correlation is that the HFA crawlers are mobile during time periods where the trees are producing a lot of terpenes. So I think it's likely that they're queuing in on these signals and are using them to find their host trees. In some of the gardens, so this is where I say if you're planting trees in an experiment, a long-term experiment, please consult a plantation geneticist or somebody that works with plantations of trees, because only one of these gardens, of the 8 gardens, only one of them we do quantitative genetics. Ben Smith was trained as an engineer. He told me that their instructions were just to plant trees, but he said he was too neurotic to just put them, so he put them in the grid. And because he planted his trees in the grid, we're able to do genetics on them, which basically means we're able to control for this E here. So this E is the contribution of the environment. So we're modeling HWA counts as a function of the terpene profiles that are there, the environment, and then a grouping effect of the genotype of another tree. And that genotype is not sequence data, it's just a name of the tree. But without having them planted in a regular grid, we wouldn't be able to control for the environment. And we wouldn't be able to fit these models. So when we look at total terpenes, we fit one model for every single month. We see that there is a negative effect of total terpenes on HWA counts. So we have a negative correlation coefficient here. When you increase your total terpenes, you're decreasing your HWA counts. So again, very different from the multi-species model, but it's what our data suggests are happening. Of course, we have 10 terpenes that are available. So the first thing I thought of was, well, let's just fit this model for every single terpene. And the literature suggested that boron acetate is an attractant for pests, and that campine and humulene may be deterrents. Unfortunately, we didn't get that pattern. So in some months, if you just focus on boron acetate, on some months it has a positive regression coefficient, so it's promoting infestation. Other months it has negative values, so it's inhibiting it, which is not what the literature suggests. Humulene kind of goes up and down. Some months are significant, some months are not significant. So when I look at these data, I'm thinking that these aromatic terpenes are not doing anything individually, but they're part of a bouquet of scents that the tree is producing. Just a return here to terpene patterns across the species. You can see that, so this is for boron acetate. You can see it's a large producer of terpenes, of boron acetate. We've got these three peaks that are happening here, two of them in the HWA crawler stage. All of the other, so these plants come from the arboretum, they're all growing in the same place. The environments aren't drastically different. The other hemlock species are generally low producers, with the exception of chinensis or caryophyllene. So chinensis is producing a whole lot more caryophyllene than other species are, this just gives us some kind of a thought that maybe there was something going on with these different terpenes, what we need to do is more control experiments to nail this down. I'm also really interested, so that cycad pollinator talk that I told you about moments ago was a professor at UGA who works on insect signaling, and after the talk, we talked, and she gave me some great ideas about setting up experiments where you are trying to attract adelgids into little traps and change the different concentrations for different types of terpenes that are there. Just try to get an understanding for how these molecules may be acting as signaling molecules. So that would be a fun experiment. Right, so those experiments with in the bulletproof stands, the Kinahan stand, there's 8 different common gardens. There was an interesting effect that was present there. So here, this is model output of HWA count. And I modeled HWA counts as a function of the tissue source, the environment, the row and column information, and then the grouping effect of the multiplier. And you can see that there's a really big effect of tissue source on the HWA counts. So the trees that were created through, that were cloned, right, they were cut the branch, you root it, you put it in soil. Those had many more adelgids than trees that were from seed and grew their own roots. So if we're thinking about doing searches of lingering trees, and we're going to go back to that tree, you're going to make a cut and do an experiment, don't do that. Because you're going to get this effect that's present there. When we include if it's a bulletproof tree or not in the model, it's not significant, and the tissue source effect is competing and displaying much more variance than whether or not it's a bulletproof tree or not. So this is actually the biggest effect from the experiment, not terpenes, not anything else. It's how did you derive the tree? So that may be a bit of a bummer in terms of this experiment, but it gives us a good way to induce an effect in an experiment. So I'm kind of optimistic about our ability to go into stand, work with mothers, work with offspring, create differences, and try to study. Okay, so our fourth aim is thinking about climate adaptation and seed banking. Really important things to be thinking about for the future conservation. So I added this slide in here this morning about population So I think it matters because not all populations are equal. Not all populations are capable of providing adaptive genetic variants that you can work with or that you can do something with. So this is a moderately popular figure produced by Hamby and Pettit. It's important in the landscape genetics literature because it decomposes how different populations have different genetic phenomena going on and what to expect when you go into different populations. So let's just focus here on this kind of contiguous range part. So imagine the eastern hemlock or spruce or an oak species or something. You have northern populations, you have kind of a range core in the middle, and you have southern populations that are there. That contiguous range, that range core area, is going to have a lot of genetic diversity. It's going to have a larger effective population size, which I'll talk about in a moment. But those contiguous range areas, the admixtures of areas, you would predict that those populations would have the greatest amount of genetic diversity that is potentially available to solve the problem. So that's one reason why population genetics matter. And thinking about effective population size. So if you go out and you count trees, that's a census. The effective population size is kind of a different definition of what a population is. And it is the size, so that the biological definition is it's the size of an idealized population that has the same amount of genetic drift as an observed population. That's a little hard to swallow. So what I tell people is that it's the number of breeding lineages in a population. So if you have more independent lineages that are capable of contributing offspring, then you're going to have a larger effective population size. This is a very important parameter to think about if you're interested in population genetics because it scales positively with the rate of adaptive sequence evolution. And adaptive genetic variants, adaptive sequences are sequences contained within genes or along chromosomes where one sequence increases the fitness of an organism relative to its population. So there are very likely going to be sequence variants that are in the range core of the eastern hemlock that we want to find. And so the range core, as we presently know it, is Massachusetts, New Hampshire, Vermont, southern Adirondacks, places where you have a lot of hemlock trees. So although we haven't necessarily found great resistant candidates yet, I think there's a lot of hope for doing that. And that's going to be an outcome of the work, this work. So the landscape genomics component, I've been collecting tissue for about a year and a half now. 60% of that tissue was collected by other people. And Tamara has helped me a lot with outreach to various groups. But we're using which is an app that allows you to collect images and metadata of the sessions. And that metadata is uploaded to the cloud. I can access it. I send people collection kits. And the collection kits have a little tree tag, some silicon gel, some basic stuff. And then people have been sending me samples. So you can see that most of the range is fairly well, except for Vermont and Maine. So the Vermonters, I don't know what's going on. They're definitely behind on it. But if you're interested in doing some collections for this project, I would be very happy to send you a collection kit. I'm going to try to wrap this up by the end of November, and whatever we have is what we're going to do. So this is the part where I showed you my great sequence data. Unfortunately, the hemlock is doing its best to be difficult, and we sent out samples to 2 different labs. One of them was a core facility in NC State, and they work with pine species all the time. They couldn't extract the DNA. Yukon core couldn't extract the DNA sufficiently to do sequencing, so that fell on my shoulders. And this is what I've been doing the last few weeks, is figuring out, optimizing the extraction method for the hemlock, getting good DNA finally, and basically it's up to me to do the wet lab work, which stinks. That's how it is sometimes. So that data is forthcoming. But what we want to do is to try to find regions of the genome that are adapted to climate, that are unique in their spatial distribution, and that should be seed banked to conserve the species. So this is the result of what a climate adaptation analysis can look like. So this is an adaptive gradient of the probability that a locus is going to be adapted. And it's related to the underlying population genetic diversity. This particular species is the red spruce. So you can see that there are some areas where there's differences in the adaptive potential of the genomes that are there. Now we want to do the same type of analysis with eastern hemlock, and then use that along with pedigrees to inform our seed banking. Very large species, we can't take everything, limited effort. So our idea here is that we can use the sequences we're collecting to identify family structures across large landscapes, and then be smart about which trees we return to for seed banking. So those are future directions. As I mentioned, a lot of this work is within the Trees in Peril consortium. That is Nature Conservancy is kind of organizing it, and there are several different academic labs that are working within this consortium. It's been really fun to work with this group of people. Some of our work has received a little bit of press. So this is an article written by Olivia Hall, the Hemlock Restoration Institute, published in Concord Quarterly. So it's been fun to see things coming together. The future directions for the hemlock, I'm actually very optimistic about it. I live in Georgia, and there's dead hemlocks everywhere, but I feel like there's a lot of potential with the work that we're doing, and there's a lot of potential for identifying beneficial trees and then doing the breeding that's there. One thing that we really need are common gardens. There are only a few common gardens of this species, and there are no range-wide common gardens. So getting seed is very difficult. There's a narrow window for collecting seed. There are easy ways to collect seed where you put a pollination bag over a branch and you just wait until it's full and then come back. But creating the network of people to do the collecting is not something I'm able to do right now, or other people have been able to do. Hemlocks are just difficult in general. They have low growth rates, low seed set. Finding sites for gardens can be difficult when you're in a ephemeral postdoc. So we're also going to be doing more chemical ecology studies and trying to understand mechanisms of tolerance and resistance in the trees themselves. So I've heard some people talk about seed banking and climate smart science, and that is at the forefront of our minds in this conservation program. Yeah, and I think with that, I'd just like to thank many partners that have helped us. We in the Nature Conservancy have been really important for bringing this project together, and folks at these other organizations. So with that, I'm happy to take any questions.
We have some time for questions.
Sure.
The Arboretum or the public garden, you said you first located that in Virginia.
Maymont Park. Yeah, it's a city park now in Richmond, and they have their own kind of house type thing there. It's just a huge manor, and then the family would move to the city.
I couldn't hear what you said originally. I thought maybe it was Louis Nature.
No, not that. It's a slightly different part of town. Sure.
Is there any sense of how diverse the HWA is? I mean, is you stated from North Carolina versus Catskills, exactly the same or?
Yes, so that is a component of a grant that we're putting together right now. We don't have the answer for that, but we want to, so there is the same species, Adirondack Invasive, is in the Pacific Northwest, but it doesn't cause massive problems there. But it will grow in hemlocks, so it's there. And it's thought that may be a natural introduction through the border forests that are there. It was introduced a few times, but in one site in Virginia, and it's spread there. So just thinking about and it also does not have sex in Eastern North America. So it clonally propagates. So it's suspected that there's less genetic diversity that's there. Work in beetles and in other invasive species has demonstrated that once you have an introduction, you'll frequently get a difference in the epigenetics of that species that allows for differential regulation of gene expression. And so the epigenetic profile of these different populations may be very different. Now, we don't know that, but that's something that we're very interested in doing. I don't know if this is fortunate or not, but a lot of the samples that I'm collecting for the range-wide hemlock study have diligent on them. So we already have those samples ready to go. We're just looking to get things together so we can work on that. Thanks.
So here in the Adirondacks, we're kind of on the leading edge of the introduction. We have lots of people who are helping us out to look for homologically intelligent, we're looking for the insect on the trees. What should we start looking about? We're looking at the trees, we're looking at the forest, to hopefully help people identify what are the patterns that we should be seeing. Like, trying to find these, maybe there are some, we have high diversity hemlock in here. What would we see on the landscape about trees that
Yes, so there's this concept of the lingering tree that is important for finding trees that may have resistance loci. Basically, if you go into a stand and you count 20 stems and 19 of them are dead, that one is alive, we would classify that as a lingering tree. And so if you're in that situation, you want to get out your tree snap app on your phone, and you go to the hemlock tab and you say, I'm making a lingering tree observation. And then a series of questions come up, you take a photograph, all that information is sent into the cloud, the photograph is georeferenced by the iPhone. And then we can go back and look at those trees and potentially collect seed or collect DNA or just do other ecological studies. So tree snap is an important way to try to get that information.
And sorry to do a follow-up question. In other landscapes where people have already started to identify those lingering trees, any idea about the timeline? Like, let's see, maybe like 5 years, 10 years, 20 years into an invasion? Like, when should we expect to start to be able to
identify that? Okay, so I don't know the answer, but that won't stop me from speaking. I think you could probably pick it up 8 to 10 years, 8 to 12 years, something like that. The Adelgid showed up in the Southern Appalachians about 2010, and it's just complete devastation. So, a decade or so later, we're able to pick it up pretty well. And there are lingering trees. So I told you about the hotspots and effective population size and genetic diversity. The largest one is the New England area. But the Southern Appalachians also has a high genetic diversity. And then the UP in Michigan also is kind of a hotspot of genetic diversity. So, so there are places where the Adelgid has been established and do these kinds of searches. And we are finding lingering trees in the Southern Alps. Yes, absolutely. So that's where part of the experimental design challenges come into play. The collection protocol that some colleagues were working with is to get cuttings of those lingering trees so that you have a clone. And I am constantly telling them, you need seed. Otherwise, you're going to be looking at a different effect. But those are things that we're thinking about. There is, so the Agricultural Research Station in Beltsville, Maryland, has a bred population of Chinensis and Carolinea, and they'll make F1s. And those F1s have intermediate Adelgid levels. All of that work has been published with the Adelgid phenotype. But we want to go back in there and look at the turkeys, because that's a great environment. The ARS knows what they're doing. They planted it in an actual garden with blocks, so you can really learn genetics. That's something we
one more question?
Yeah, so ash is one of the groups, one of the species complexes that is being worked on in true species encounter. That is, who's working on it?
Most of that's happening out of Holden, and it's further along in that they have identified resistant ash, and they're already up to grow out some resistant ash from seeds, and they're much closer to commercially available.
That's right. And Jennifer Koch is kind of leading a lot of that work, who's in Delaware, Ohio, at the Forest Service. All right, thank you so much.
Great, everyone. Well, we can't tell you how much we appreciate everybody coming and spending their day with us here at IGNIT and all the different speakers. Hopefully you learned a lot. You learned both about the challenges that we face, but then also the great opportunities that we have to use new technologies, reach out to these incredible researchers from different organizations, different universities, and be able to tap into and know that we have some of these resources through there. So, Sean, I don't know, do you have any housekeeping or things? Oh, I'll let you say that before I do our last couple of things.
Just want to let everyone know, well, first of all, thank you for being here. It's a great crowd today. But second of all, there's a ton of ribs and stuff left in there, and there are takeout boxes, so please don't be shy. Go help yourselves. Take as much as you want. I would hate to throw it away. Thank you.
Yes, well, back up, we have all sorts of, materials and handouts. Make sure you sign in on the sign-in sheet today. If you haven't joined up as part of our newsletter or bring your email, please keep track. We will send out some resources to all the people who registered on here. We were trying to record the talk, so hopefully that worked out well, and we will be able to post them online. And